Okay, guys, welcome back. We are on and live from ICTS. This is Gabriel Mindlin's second talk. So here is Gabriel Mindlin live. Go ahead, Gabriel.
Okay, well, thank you very much, Ram. It's a pleasure to be with you people together again. I hope to meet you sometime in person soon or sometime. So, what I'm going to be talking about today is, in principle, this general idea that we have in the lab, which we push through very different projects, which is the following. We are interested in neuroscience. We do neuroscience measurements, as I was telling you before. We put electrodes in the brains of birds and try to unveil how behavior is coded in different parts of the avian brain, how learning takes place. But something that I'm particularly interested in, and this is the reason why I, as a physicist, started to work in birdsong, is that behavior emerges from the interaction between a nervous system, a biomechanical periphery, and the environment. It is not the nervous system just driving passively some robot or something. So one thing that I'm very interested in is how the biomechanics, and in particular the nonlinearities in the biomechanics, condition some behavior. So what I decided to do today is to put in the presentation two examples, and I will kind of go through them more or less quickly, and if you're interested, we can discuss in the breakout room session. And one question is, at which rates do birds breathe? So breathing is a behavior that requires the control of the respiratory system, that is controlled by the nervous system. And why do birds breathe at 2 hertz and not 5 or 1.1 hertz? Okay, what conditions the rhythm that is behind that? At what rate do they sing? Why do they sing with faster frequencies while we as humans speak with lower frequencies than our breathing frequency? And then a second example is how much of the acoustic is determined by the brain and how much of the acoustic is determined by the biomechanics driven by simple instructions. And those are the two examples that I decided to share with you today out of the many ones that we do in the lab, focusing on that particular question on how the biomechanics conditions behavior. So I'm going to start with the first one, breathing. And as I was telling you before, in all the avian brain, we have these nuclei in the brainstem, which are RAM and PAM, which generate the oscillations that are involved in the control of the respiration. One is controlling the expiration, the other one is controlling the inspiration. And the birds breathe, as you will see in this GIF, when air passes through these rigid lungs and is pumped by these flexible air sacs, okay? So the complete circulation of air throughout the respiratory system takes 2 motor cycles. It's kind of complex. So what I'm showing in the GIF is 2 paths of air. One is the brown one, the other one is the blue one, and I use that as a pedagogical tool to try to explain what happens. So what happens in the respiration of the birds is that in the first expansion, so I'm going to be describing 2 cycles. Now, in the first expansion, the air enters and goes to the posterior sacs, and the air that is in the lungs goes to the anterior sacs. And in the contraction, what happens is that the air in these sacs go to the lungs, and the air that is in the anterior sacs gets expelled. So you need 2 cycles to complete one respiratory cycle, okay? And this mechanism guarantees that oxygenated air is passing always through the lungs. It's a very efficient respiratory dynamics. So the expiration and inspiration are controlled by the neural nuclei in the brainstem that I was showing you before. And the question is, why do canaries breathe at approximately 2 hertz and not 1 hertz or 5 hertz or 10 hertz or 0.1 hertz? So the first thing that we did is to build a biomechanical model for the respiratory system. So we're going to have a cylinder with 3 subdivisions. The lung is represented by this part of the cylinder, and these 2 parts are going to be representing the 2 sacs. And as always, I try to measure what I'm talking about in terms of a model. So what I measure is, as I was telling you before, with a pressure transducer, which is the pressure here. And what I do is I build a device with the whole transducer. I place here in the ribs where you have the sternum, okay, and the ribs finish here. I place a whole transducer, and then I glue a little magnet in the abdominal. So when the bird is moving the abdominal thing, what I get is a time trace that describes the volume of the air sacs. So I measure simultaneously the pressure and the volume. And the reason for which I do that is because the lungs are not close back. So if you measure just the volume, you don't have the complete information you need in order to find the pressure. It's an open cylinder, okay? So you have these two valves that correspond to the valve of the posterior sacs and the valve of the anterior sacs. And this little biomechanical model allows you to represent the avian respiratory system. So the piston represents the sternum, the rib cage, and the overlaying muscles, okay? And the movement of the piston changes the volume of the three cavities, representing the anterior and the posterior sacs. These are the valves, aerodynamical valves. These are not physical valves. These are valves that, through the shape, make it easier for the air to flow in one direction than the other. So I measure the pressure, as I was telling you, with the pressure transducer. I measure the movement here with this little magnet and the whole transducer. And we can build a model for this thing. So at least for small oscillations, the first approximation will be a linear model, and you can state which is the dynamical equation for the piston. This will be a typical Newton's equation with the restitution, dissipation, and then the effect of the difference of pressure between the outside and what happens within the cylinder. And then you can write state equations to relate the pressure with the geometrical variable x. So we use just the state equation of the ideal gas, and the number of particles, the amount of fluid here is determined by the flow that is either entering or getting out of the system. In order to represent that the valve for the input can be different than the valve of the output, I use a nonlinear function. So this will be, in principle, a piecewise linear system. And then you can relate the flow with the pressure. This is like a Poiseuille law, and it's something that you can experimentally verify. There is a linear relationship when each of the valves is being used between the pressure and the valve. So S is a sigmoidal function. So when the pressure differential is positive, this will be 1, and you will have that the valve that you have to use is the anterior valve, and when delta P is negative, it's going to be the posterior one. So now we're in conditions of writing a three-dimensional dynamical system, okay, that represents the behavior of X and P, which are not in principle in phase unless these valves are almost closed, okay, and you have a closed piston. And of course, what you have is the driving force that corresponds to the activity of these muscles, either expiratory or inspiratory muscles. So you drive this system, it has a tiny inertia, but the main thing is that there's going to be, in principle, a phase difference between X and P because the valves are open, okay? So it's not a closed cylinder. So when you do that, now we have the variables of the pressure and basically the volume of these sacs. So here I'm showing you the time traces of the displacement. This is what we measure with the whole pressure, the whole transducer, and we measure simultaneously, okay, the pressure, okay, in the air sacs. And what I'm showing you in here are the time traces with points, the experimental ones, and with the lines that you have in here, the result of fitting the model. So what I'm doing now is fitting the model. In principle, for the quiet respiration, I'm going to assume that the valve that connects the anterior sacs with the bronchi is the same size as the valve that controls the exit from the anterior sacs to the bronchi. When you do that, you can fit any fitting procedure of your choice, the parameters of the model, and what you get is an extraordinarily good fitting. Now, here is something that I find very interesting. When you want to measure something in biology, if you want to deal with it like in a normal physical experiment, probably you're going to be putting your system outside its normal operative conditions. So, for example, it was tried to be measured these features of the respiratory system in a preparation. But typically you have to put in a pressurized chamber, you have to put some little rings to avoid the closing of the labia and so on. And then when you measure, which are the biomechanical properties of the respiratory system, you change your system so much that what you feed has nothing to do with the real system that is operating on life. So in this other approach, instead of measuring the biomechanical parameters of a static physical device that I place in the lab, I develop a model and I measure with minimal interventions the dynamics of the problem. I fit the parameters, and then I have a number of parameters that were fitted with the device working normally. Okay, and when you do that, you find the following, which to me it's really nice. Each of the lines here correspond to fitting the parameters of this model with a segment of the time trace, 10 oscillations, 15 oscillations, 5 oscillations. And then looking at the resonances of this system for different forcing frequencies. Okay, so I take a segment of the signal, I fit the parameters, and then I study the resonances of the system. And I do that for many segments during the day. So what you have in here is for 3 different birds. All these gray curves correspond to the resonance curve of the oscillator whose parameters were fitted by experimental data. And as you can see, they are all very similar things. You can find the average resonant curve in here. And when you look at which is the resonance of the average curve, what you find is that these oscillators have their resonances very close, extremely close to the average breathing rate. That means birds breathe at the resonance of its biomechanics. And it is interesting because it's not the only behavior that the birds can have in terms of breathing, okay? So here what we have is the distribution of the respiratory rates, and here the distribution of the resonant frequencies of the biomechanics. And those two peaks coincide beautifully. And if you look at other regimes, like for example, song regimes, the rates at which birds can produce their song can be completely different. So what birds do is they breathe at the rate at which they have the resonance of their respiratory system. That means they do it all the time. We also breathe all the time, but birds breathe all the time, and therefore the breathing uses the resonance of the system, which makes a lot of sense. So, of course, what happens when you sing, and this is very interesting because when you sing, the pressure is much higher. Okay, so here we have the normal breathing pressure, and these huge peaks are the huge peaks that I was telling you about. Each time you want to generate a syllable, you have to have a large expiratory pulse. So now the question is, when you do that, what happens with all these linear approximations of the biomechanics? And of course, the system is no longer linear when you push it beyond the normal respiratory amplitudes. And when you introduce the nonlinear restitution constant, what you find is that, so here in the restitution constant I was telling you about, we include a nonlinear term. Actually, this can be fitted with passive normal experiments. And when we take into account this nonlinear fitting, what you find is that now the system shifts its resonances and starts to shift it upwards, okay? So what I say is that when you want to push this system to higher values, then the resonances shift to higher values. That means that it also makes sense that when the bird is singing, increases the rate of production of the syllables. So now what we're doing in the lab is precisely that. We're studying the relationship between the amplitude of the forcing with the syllabic frequency in order to test the hypothesis that it is the change in the shift in the frequency, resonant frequency, what determines the frequency of the singing. Okay, so our conjecture now is not only they breathe at resonances, but also they sing at resonances. And why they have different syllables? Well, they also manipulate the output size of the respiratory system. Now, I was showing you in the first lecture that when birds want to sing, the first thing they do is they close the labia, and the labia get closer, and you can induce the oscillations. So by decreasing this aperture, you also change the parameters of the system. We're in the process of testing that for each singing frequency, the system is precisely tuning the resonance of the respiratory system. So this is an example of how biomechanics conditions the behavior of the nervous system. And I wanted to complement this vision with another example at another order of magnitude of the problem that has to do not with the slow oscillations of the production of the syllables, but the nature of the vibrations themselves and how they convey information for the brain. So what I'm going to tell you now is how much of the acoustics is determined by the brain and how much by the biomechanics. And to do so, I want to go back to this example I was telling you 2 days ago. What I'm showing you here is the biomechanical model that we produced when we started in this field. So to remind you, we have labia with a restitution constant, which is this. What I'm doing is computing the Newton's equations for the midpoint position of the labium, and we have the nonlinear dissipations, the restitution constant that depends on the elasticity of the labia, and we have the average pressure between the labia, which is what allows you to transmit energy from the airflow to the labia. What I was showing you is that this sounded pretty good to us. So if we did pass in parameter space that would reproduce the fundamental frequency, all the harmonic content looked like it was already contained in the nonlinearities of the problem. So I showed you that this model allows you to have both saddle node in limit cycle oscillations and Hoff bifurcations, so you can produce either tonal sounds here or sounds with rich harmonic content, and that behavior can be captured not only by this very simple biomechanical model, but by the normal form that corresponds to further simplifying this model, eliminating the non-resonant terms and just finding which are the minimum nonlinearities that are consistent with the dynamics. And you have the same behavior, and you can now synthesize sounds not only with the biophysical model, but with the dynamical model of the normal form of that system. So it's minimum simplicity, maximum simplicity, minimum complexity, and all the information of the path in parameter space, like the pressure fluctuations that allow you to go to this phonating region, returning or increasing the fundamental frequency, is captured by two simple parameters. Okay, you put the time dependence on these parameters, and supposedly you can generate good songs.
What is gamma?
So gamma is a scaling
scalar.
It's a scaling constant that would allow you to have the right harmonic content at the right frequency. So that is not changed for a particular bird, actually for a particular species, but different species can have a different content. So let's say if you want to have something with a high spectral content at a particular frequency, you control that by the gamma. So we fix gamma for the whole species, and we don't touch it, and then the only things that we change in time are alpha and beta. So we wanted to test this model, besides, or beyond giving rise to more or less decent songs. One thing that we did at the beginning was to try to see whether we could capture something interesting of this song by this model. So let me tell you something about the zebra-finch song. You can have very rough sounds, okay, spectrally very rich, like this one here, or you can have almost tonal sounds, okay, spectrally very pure, like these high-frequency notes. And one very interesting thing is that if you compute the spectral content index, so that means how many harmonics you have, okay? This is an index that tells you how rich the spectrum is. It's going to give you a high value if you have many peaks of harmonics, and it's going to be giving you a value of 1 if you have just a pure tone. So if you compute for different syllables of different birds, the relationship between the spectral content and the frequency, what you find is that all the birds and all the syllables can be fitted by almost a one-dimensional curve. That means that when you have higher frequencies, they're always tonal. When you have low frequency sounds, they're always very rough. And this seems to be universal. We know all the time we're adding points in here. This was the first one we did, but we keep all the time adding points in here. It's like almost the first exercise that the student does, contributing with some of this analysis, and the fitting is extraordinary. And now the curve that I'm showing you here is what you compute when you do this calculation with the basic model. So that means that in this model, when you are approaching the saddle node in limit cycle bifurcation, you have spectrally very rich solutions of low frequency. And then you're having this part of the curve. And then when you are moving away, what you have is that the sounds become more and more tonal and spectrally very poor. So something very basic was captured by this very simple model. So as I was telling you in the other class, you can get more or less decent copies of the song. So here it is one example. Okay, and here is the normal form singing. Okay, and we wanted to test that now by exposing a bird to its own song. And what is the neuroscience experiment here? It is the following. There are neurons in this part of the brain, which is part of the telencephalon and part of the motor pathway, which are highly sensitive to its own song. That means this part of the brain is generating rhythmic behavior that controls respiration and attention, but also is receiving information from the auditory pathway. Okay, so here there is an integration between the generation of the song and the feedback that the bird is listening to. So there are neurons that are highly tuned to its own song. The bird is programming itself to expect some outcome of its song. So there is a mapping from the auditory pathway to the motor pathway. So when the bird is asleep and you play its own song, the auditory neurons trigger the activity of the motor neurons and you see activity. The interesting thing is it's extraordinarily tuned to its own song. So if you play the bird something else when it's asleep, there is going to be no response. No response if you play the song of a canary, this is a zebra finch, and not response if you play the song of another zebra finch. Only if you play its own song, you're going to have a response. It's this mapping from the auditory pathway to the motor pathway that gets activated. So let me show you an experiment. This is a bird that is singing, and you're going to be listening to a chirping that corresponds to the spiking neurons in the HVC activity. This is while the bird is not singing. Ram, just one little, I know that you hate if I say that, can you listen to this, but can you listen to the sound of the movie?
Yes, yes, yes. We
Okay. Okay, that was important because So when the bird is when you see that the bird is singing, you will see a modulation of the spiking. Okay. Okay, so there is this homogeneous activity when the bird doesn't sing, and then there is this modulation, which is coding, in some mysterious code, the behavior. So now, what happens when the bird is sleeping? So now the bird is going to be sleeping, and from time to time we're going to play its own song. And I want you to listen what happens to the brain activity when the bird is listening to its own song. Okay, so you see, and when you compare the pattern of activity when the bird listens to a song, is identical to the pattern that is used when the bird is during the day producing the actual song. So, so what we decided to do is to do this little experiment of exposing a bird to its own song, okay? And we see here the huge activity that is induced. So in this panel down here, I'm showing you a point per each spike that is being recorded. And in the vertical axis, what I do is I superimpose the result of many different experiments. So you see this clouding here because each time that we play the song, there is a higher level of activity. And now I add all this together, and this is what I'm showing you in this panel. So again, when you play its own song, we have this huge response. And when we play the reverse song, that means the same notes, but in a reverse order, okay? Okay, when Ram, Neelima, and I were younger, okay, there was this myth that if you would play the records, okay, in reverse, you will hear, hidden messages or something like that, right? So well, this is exactly the same. But the bird doesn't respond at all, okay? Doesn't respond at all. Okay. And now if you play a conspecific, that means a neighbor, okay, that is singing, there is no response at all. So the question was, well, now we use the normal form, which is this beautiful, simple model, okay, that the physicists like, right? Because it's a dynamical model, it's very simple. We're going to find what? Some response. And when we did the experiment for the first time, we found nothing. Now, listen how unfair this is, okay? This is the Birdsong song, and this is the synthetic copy. Now, and the bird doesn't care at all, okay? So it was devastating. And I was in Chicago at the time. I went there. My colleague, Dan Margollia, she's, one of the most renowned neuroscientists in the field. I was explaining that the physicists can, do magic with simplification of the biomechanics. And then when we did this experiment, I didn't know, I just wanted to vanish, disappear magically, just be teletransported to another planet or something, because it's the most incredible failure that you can have. Not a single neuron would spike, okay? Okay. Now, this was very interesting because when you do physics and you do a simplification that doesn't work, usually you know what you have been oversimplifying and you know which is the order in which you have to correct your model. When you are working in biophysics, you don't know. What is important for a bird? What didn't I include in the model? Why was that so important in evolutionary terms? So those were questions for me interesting enough, which is the hierarchy of importance of the different parameters that allow you to do a sensible simplification from the physical point of view. Can you do the reductionist approach of physics to neuroscience and biology? So, like in many cases, you can be smart or you can be lucky. And in this case, I think we've been lucky because we improved the model in one direction that was one among many. What we did is we introduced some more detail of the filtering, which we had oversimplified, and we introduced some noise in the parameters. And I want to go back to that because it's related to something you were asking, Ram, 2 days ago, when you were talking about simplified models or more complex models. We needed to have noise in the simulations, or at least I tried, because that will take away some synthetic aspect of the song. And now when we did that, what we found was now responses. So it was magical. We had to add some noise in the parameters and a little bit of the filtering, although now we're doing some experiments with Anna and ruling that the filter was so important. But we added these two things together and magically the solutions jumped from nothing to something very decent. Okay.
Question,
now,
yeah. Quick question. If you take the original, the sound, the one on the left, and smooth that is, remove some noise from that,
right.
At which stage does it stop responding?
Oh, well, okay. So let me tell you one thing. This is noise that you add to the final sound, but noise that you add to the parameters. So you cannot just filter the sound. But we did exactly that question on how smoothly, okay, this appearance is. So what we did is the following. We did exactly what you're suggesting. We had in one axis different levels of noise. In another axis, different levels of dissipation of this upper track that I was now introducing. And what we did is a network of experiments where what we did is expose the bird to different versions of this, which of course to me sound all more or less the same. And we exposed the bird to all these different matrix of things where we were changing the noise and changing the dissipation and look at the neural response of the bird to this thing. So what I'm doing when I change the noise is not the noise added to the sound, but the noise added to the parameters that are used in the model to generate the sound. And when we did that, we found this beautiful surface where there is an optimal level of noise where the bird responds. So if you start with no noise, you have very poor responses. Then you start to increase the noise and you get to maybe 70% of the responses. We're getting now, this is the first work we did. This was like, I don't know, a few years ago. Now we get better responses, but this was about 70% of the response. But then if you keep on increasing the noise, then what you're going to have is a decrease in the response again. And this to me, it's very interesting from several points of view. One of them is, what does this level of noise tell me? And I think it tells me which is the limit of what an oversimplified dynamical system can give you in terms of the control parameters that you use. If you remember my first talk, I was showing you how similar the pressure gestures were, and I could have done the same with the tension. Those are the output of a low-dimensional dynamical model. Now, if you take a higher dimensional dynamical model, like, for example, modeling many neurons, like phase oscillator or something, maybe the average can be similar to the gestures that I have, but when you take the sum of all this activity and you project it to the masses, you're going to have fluctuations because you don't have 2 or 3 equations controlling the average. You will have the average over a few thousand neurons and you will have fluctuations. I think that the noise that you have in the parameters that you need to have a realistic response is related to the fact that you need to take it into account a higher dimensional model and do an average instead of just finding a very low dimensional model for the nervous system. So I think this is a very interesting experiment in order to test the hypothesis of how realistic can be a very low dimensional model in neuroscience. And I think that, clearly puts a limit, and quantifying the noise allows you to have an idea of how detailed your model should be. No, I think it's a very interesting thing. The other thing that to me is interesting is the following. And this is, if you want, a cultural question that I have, a general cultural question on the relationship between physics and biology. By doing these kind of experiments, what I can do is take a model which is more or less successful. I don't know, one that can elicit 70% of the response of the real song, and start to play with the different parameters of the model and find which are the parameters that induce a low decay in the response against those which induce a high decay of the response. That means to have a hierarchy of importance on the different parameters of the model in order to induce a realistic response. And I think this is evolutionarily important. Some things are neutral, some things birds don't care about. But do they care about the frequencies that indicate the size? Is it more important to miss that variable? Does the bird really expect to have a good reflection of its own body? Do they pay attention to clues that give you information on the size? Or the precision? Is the temporal precision? Is the level of noise? Which are the variables that are really important to the bird? And I think you can do these kind of experiments in order to state the hierarchy of importance. And I think that it's very important when we do modeling for neuroscience or biology in general to have this map, the hierarchy of importance of different effects. Because a biologist can tend to, in my lab, half of the people are biologists, and they have this rapid response. Everything is important in biology, right? Because evolutionary took you there. Everything is important. I say, well, okay, I cannot work with that. Is there a hierarchy? I mean, what is more important? Well, I think this kind of experiment can take you in that direction. So let me go to the conclusions of this. And in general, the idea is that, yes, we do find that neuroscience and biophysics and the body and its nonlinear features are strongly each other. So birds, for example, breed at resonances of their respiratory biomechanics. Okay, they have this. They have specific frequencies. And they are conditioned by the biomechanics. And the spectral features of sounds, for example, strongly condition dynamics of the biomechanics, okay? This is the secret behind the spectral properties of sound. And both examples illustrate the deep relationship between the nervous system and the biomechanics at the origin of behavior. I think it is very important not to address the problem of neuroscience just like it's some dynamical system that drives passively a body. I think that they are strongly interconnected, and we, as for example, the ones that come from physics, have the special responsibility of pointing out to how biomechanics and physics and dynamics condition also behavior and the brain activity. And this is the story today. I think I did it on the right time, Ram. Thank you very much.
Absolutely, it was perfect timing. Before I open it up to everybody else, let me sneak in a question. Some years ago, there was this. There was a lot of work on allometry, the relationship between the metabolic rate of a species and its size. Is there an allometry in, let us say, the frequency, the syllabic frequencies of your birds, the size and versus the size? Because one would feel that there has to be big birds will have different sounds from small birds. But is there some work on this?
Yeah, actually, I don't want to self-advertise a lot, but we have a physical review letters last year on exactly that subject, Ram. It's exactly on that. Because, and let me tell you what the question was. We have to tell people that we didn't rehearse this, right? Because the question that I had was very, very similar. It's like, because the question was this: since many of these species learn, they can do something like to trick others and cheat by changing its vocalizations in order to sound larger than they are. They can do that. So the question that I have is like, is there any kind of signature that is honest in the song about this? Is there an honest signal in the acoustic messages of the size? And in many species, there is not, because they can cheat. When you learn, they can cheat. So what I did is I took another species that is Phytotoma rutila, which is a Sabocine bird. And that Sabocine bird, they cannot cheat because they cannot learn. And also they cannot cheat because this is what happens. They produce with their vocal organ very, very, very spectrally rich clicks, right? It's like a click. And if you go to the webpage of my lab, you have movies there, and also there is a link in the publication that was in Physical Review Letters with the movie. It's a bird that sounds really strange because it sounds like clicks. And then the click is filtered by the oropharyngeal cavity. So they have something that sounds like when you are opening a door, okay, after a long time. It's like a very strange sound. But since you have these low frequency sounds that are spectrally very rich, and then you have a filter at a few kilohertz, so the clicks are produced at, let's say, 100 hertz with high spectral content, and then you have a 4 kilohertz filter, what you have is a band that is associated to the size of the oropharyngeal cavity. Now, if you do a scaling analysis of this Helmholtz resonator, you know that the frequency then scales specifically like 1 over the size of the bird. So what we did is we took recordings from Argentina to Bolivia of the songs, and then we measured specimens, the femur size of specimens from a museum of birds that were taken from Argentina to Bolivia. And from Argentina to Bolivia, there are like 4,000 meters of change in altitude, and the birds get larger there. So although we didn't weigh the birds, we knew that the birds were larger in Bolivia than in Argentina. We tested that with the femur sizes of the specimens, and we compared that with the frequency. And there was a scaling that was exactly like you predict by the allometric measurement. So there is an honest signal in some species of birds. So birds that don't, so concretely, the answer to your question is birds that don't learn are obeying these allometric relationships, and birds that learn skip it and learn to cheat. So the first, one of the first things you do when you learn is to learn to cheat, okay, and send a message.
Okay, good on that one. Let's move on. Questions, everybody?
So, with regard to what we were discussing yesterday, some of these features should show up in our analysis, right? So, what would you expect?
I think I didn't get to Neelima.
You can hear me?
Yeah, but I heard you with some resonance effect. And
and also the time lag. So, okay, let me just try once, otherwise we'll do this in the chat room. See, some of the things which we discussed yesterday, namely, what is the relation? They should, see, what we are talking of is the relation between the biomechanical system and the sound, right? So, in fact, what we were looking at yesterday, namely the amplitude envelope and the sonogram. So, some of these things should show up when we do the comparison. So, what would you say is a good candidate to look for?
So there are. I think that this principle applies to every scale. So, for example, when we are talking about sound amplitude, we are talking about pressure, and we are talking about how the respiratory system is conditioning the lower time scales of the problem, which are the syllabic production. And then you have the higher.
Could you speak a little louder, Gabriel?
Yeah.
Yes, could you just speak a little?
Yeah, so what I was saying is that the amplitude fluctuations are related to the respiratory rhythms, and those are the low temporal scales of the problem. So how the biomechanics conditions the sound production at the low temporal scales is related to how the respiratory system conditions the brainstem. And then you have the higher frequencies, which are the frequencies of the labial oscillation. And in that case, the conditioning comes in terms of the nonlinear dynamics of the labia and how they condition the acoustic features that you generate. So I think that at every temporal scale, there is a condition of the biomechanics to the nervous system. The lower time scales, the amplitude that you were mentioning, related to respiration, and the other one, the passive motion of the labia for the faster. But in both cases, there is a conditioning effect.
Thanks.
Perfect. Okay. Thank you for your